Hello everyone. DNA is very long molecule in a typical human cell. The DNA double helix has about 6.6 billion base pairs. Now let's do a simple calculation. Distance between two base pairs is about 0.34 nanometer. That is 0.34 multiples 10 power minus 9 meters. Total length is 6.6 multiple 10 power 9 base pair multiply with 0.34 multiple 10 power minus 9 meter per base pair. Now the result we get is 2.2 meter. Imagine 2.2 meter of thread-like DNA is stored inside a nucleus that is only about 10 power minus 6 meters or just equal to 1 micrometer in diameter. इतनी लंबी DNA इतने छोटे न्यूक्लियस में कैसे समा सकती है? That's exactly what we are going to learn today. DNA packaging. DNA packaging is a fascinating and efficient process. Let's take the example of prokaryotes like E. coli. The DNA in E. coli is about 1.36 millimeter long. Let's calculate how many base pairs that is. 1.36 millimeter just equal to 1.36 multiple 10 power minus 3 meter. Number of base pairs are 1.36 multiple 10 power minus 3 meter divided by 0.34 multiple 10 power minus 9 meter. We get 4 multiply 10 power 6 base pairs or 4 million base pairs. लेकिन बैक्टीरिया में तो न्यूक्लियस ही नहीं होता. Exactly, but their DNA is not floating randomly. In prokaryotes, the DNA is localized in the region called nucleoid. Here, negatively charged DNA is held together in large loops by positively charged proteins, forming a compact structure. Packaging of DNA in eukaryotes. The histone system. In eukaryotes, DNA packaging is more advanced. Let's look at the role of histones. Histones are positively charged proteins. They are rich in basic amino acids like lysine and arginine. These amino acids carry positive charge which attract the negatively charged phosphate backbone of DNA. 8 histone protein come together to form a unit called histone octamer. DNA histone octamer के चारों ओर कैसे लिपटती है? About 146 base pairs of DNA wind around each histone octamer, forming a structure called nucleosome. Histones are special proteins loaded with positively charged amino acids like lysine and arginine. Since DNA's backbone is negatively charged, they stick together like magnets. There are 5 main types of histones: H1, H2A, H2B, H3 and H4. A nucleosome core consists of 8 histones, 2 copies each of H2A, H2B, H3, H4, forming a spool around which about 150 base pairs of DNA wrap nearly 2 full turns. Why wrap DNA? By wrapping DNA around nucleosomes, the length is reduced to about 1/3 of its original length. Still too long for the nucleus, but it is great start. Histone H1 is like a click that locks DNA in place on the nucleosomes, helping seal the DNA spool and stabilize the structure. Packaging DNA tightly is essential, but it also poses challenge. DNA needs to be accessible for copying and reading during cell activities like replication and gene expressions. The nucleosomes and chromatin structures strike a delicate balance between packaging DNA efficiently and keeping it accessible. In short, DNA wraps around histone proteins, forming nucleosomes. Nucleosomes fold into thicker fibers and then into chromosomes. This incredible packing allows meters of DNA to fit inside a tiny nucleus, all while keeping it ready for life's most important process. And 54 base pairs of linker DNA, that is 146 base pair of DNA wrapped around it and 54 base pair of linker DNA. A total 200 base pair per nucleosome unit. तो न्यूक्लियस में कितने न्यूक्लियोसोम्स होते होंगे? In a single human cell with 6.6 multiple 10 power 9 base pair of DNA, number of nucleosomes are 6.6 multiple 10 power 9 divided by 200. We get the result is 33 million nucleosomes. Nucleosomes are the first level of packaging, but during cell division, DNA is packed even more tightly. Nucleosomes fold into 30 nanometer chromatin fibers. These fibers further coil and loops. Eventually, at metaphase, DNA is condensed into visible chromosomes. This higher level packaging is supported by additional proteins called non-histone chromosomal proteins (NHC). These proteins act like scaffolding, helping the chromatin loop and fold even more. Chromatin is not uniform in all parts of the nucleus. It exists in two forms, euchromatin and heterochromatin. Euchromatins are loosely packed, stain, light, and transcriptionally active. Genes can be accessed easily. In heterochromatin, they are densely packed, stains dark, and transcriptionally inactive. जब सेल काम कर रही होती है तब कौन सा क्रोमेटिन एक्टिव होता है? जब जीन्स को एक्सप्रेस करना होता है तब यूक्रोमेटिन एक्टिव होता है. Let's summarize. DNA is very long, but it's tightly and efficiently packed. In prokaryotes, DNA is locked and anchored in the nucleus. In eukaryotes, DNA wraps around histone octamer to form nucleosomes. Nucleosomes fold into chromatin, which further condense into chromosomes. Two types of chromatin, euchromatin, which are active, and the heterochromatin, which are inactive. Can you answer the questions? Can you explain how DNA negative charge help in its packaging? What would happen if histone proteins are missing? And last, why is euchromatin more important for gene expression?